diversity in male and female songbirds, a case study from the honeyeater family that will be presented by Michaela Roper from Massey University in New Zealand. So, hi, Michaela.
Hello.
Can you share your screen, please?
Yes.
Thank you. So you can go ahead and present when you are ready.
Thank you. Hello, everyone. Thank you for coming to my presentation today on understanding the link between morphology and vocal complexity in male and female songbirds. Today, I'll be focusing on some preliminary results from my PhD research, which I focus on the syrinx structure of the Ocine passerines in one particular species, and I'll be continuing this for my postdoc work. So within the songbirds or Ocine passerines, we have a lot of different diversity levels of vocal complexity. This varies from simple calls to very complex song. And why we see differing complexities, particularly within the songs that species sing, is thought to arise from female mate choice and male-male competition. However, this research has been focused on species where males sing, which means that the selection pressures may be biased towards males versus females that sing, because more research is showing now that female song is highly prevalent within the Ocine songbirds, particularly in the tropics in the southern hemisphere, even more in the northern hemisphere now. And as recent research has shown that female song is even the ancestral state of songbirds. So female song is just as variable as males, with some species not having song and other species having song repertoires that are comparable to males. And so more studies are needed to compare both male and female song to be able to understand why this vocal complexity exists, particularly within songs. And to do this, I'm going to be studying species within the honeyeaters. The honeyeaters are an Australasian clade and a very old group. And they, as you can see, they vary in their morphology and their habitats, and they also vary in their ecology, life history traits. And importantly, they have a huge variation in their differing songs. For example, we have simple calls such as in the bell minor. I'll give an example. To more complex song where a species only has male song, in the singing honeyeater, for example. Sorry, let's go back. To species which also have complex female song, such as in the Korimako. So to further understand why such variation exists across both species and sexes, we need to further understand the production of song and other vocalizations, and particularly compare between the species. So what we know so far is that song production in birds is controlled by song control regions within the brain, and the size of these different regions have been linked to the song repertoire size in some species. However, this has not been supported in research that included species where females sing or sing more comparably to males. And other aspects of vocal production include quite a variety of different movements that include the beak and the trachea, and most importantly, the syrinx, which is the vocal organ of songbirds. The syrinx consists of a tympanum, which is fused rings from the trachea, bronchial half rings, particularly B1, 2, and 3, and the lateral and medial labia, and the connecting muscle tissue. And what research has been done so far has shown that there is sexual dimorphism in the syrinx size between different species. And research on the zebra finch, where only females sing, they found that the male syrinx was up to twice as large as in females, particularly half-rings B1 and 2 were larger. These bronchial half-rings are known to have some influence on the fundamental frequency that bird sings, so this led the authors to predict that the male syrinx has adapted to producing higher fundamental frequencies in males compared to females. This research was then repeated in the European starling, where females do sing, although they have a smaller repertoire size than males. And what they found is there is reduced sexual dimorphism in their syrinx size compared to the zebra finch, where the male syrinx was only 30% larger, but the half rings B1 and B2 were still larger in the male, which relates to the higher fundamental frequencies and bandwidth that the males sing compared to females. But this reduced sexual dimorphism also led the authors to hypothesize that perhaps this less size difference between males and females also could relate to the difference in their song repertoire size. So two hypotheses that came about from this research is that larger syrinx size predicts higher fundamental frequencies, and syrinx sexual dimorphism predicts song repertoire sexual dimorphism. So obviously there's only 2 species studied so far, so I wanted to further investigate these hypotheses in a wider range of species, but today I'll be only presenting it for one species, the Korimako, which is part of my PhD research, or the New Zealand bellbird. They both have male and female song, but compared to the starlings in particular, they have much more sexually dimorphic song. And while the males have bigger repertoire sizes than females, they only share about 90% of the syllables, which are the individual components of a song, making their song very different from each other. And they also sing year-round. So my questions were, how does the syrinx differ in size between the Korimako sexes, and particularly focusing on the half rings, B1, 2, and 3? And then how does this level of syrinx sexual dimorphism compare between the other 2 studies on zebra finches and European starlings? And then ultimately, how does this relate to vocal complexity and the 2 hypotheses that were drawn from the previous studies? So I collected naturally deceased specimens of the Korimako on Tiritirimatangi Island in New Zealand, where I was able to collect 1 adult male and female for the study we'll be presenting today. And we also recorded sounds of adult males and females to get their syllable repertoire size. So as mentioned earlier, the syllables are the individual components of a song, and each boxed up part of the song represents a syllable type, and each color represents a different type. So to measure the syrinx, we use microcomputed tomography, and we prepared the samples as another paper by During et al. 2013, and we dissected the syrinx out of the body with the organs intact and measured body measurements, fixed the specimens and stored them, and then micro-CT'd them at the University of Auckland, and it took about an hour and a half to scan each individual. To calculate the syllable repertoire size, we use Koe, an open source software developed by Massey University, which is freely available. And we labeled over 18,000 female syllables, 5,000 male syllables, and this allowed us to measure the repertoire size for 9 individual females, 25 males, and also measure the average frequency bandwidth for the species. So once we had the micro CT images, we then had to build 3D models, which we used a program called Drishti, developed by Australian National University, where we could select each half ring and measure its volume. And then I could compare it between the sexes with the paired t-test, and then with the other species using a male to female ratio, which was also adjusted for differences in body mass between the sexes. So here are the results for the Korimako syrinx, with the male on the left and female on the right. As you can see, the male syrinx is obviously quite bigger than the female, but overall they have the same structure in their trachea, tympanum, and bronchial half rings. And looking at the graph on the top, we have each half ring on the x-axis and volume, y-axis and volume on the x-axis. And in the black are the males, the white the female. And overall, we can see that the male bronchial half rings were larger than the female, except for one on the left, which means we didn't find a significant difference between the left side, but we did for the right side. And then to compare with the other species, I collected data on the frequency range, syllable repertoire size, and body ratios. And as if we particularly pay attention for the syllable repertoire size of Korimako and European starlings, they have much more similar level of dimorphism, with females having about half as many song types as males compared to zebra finch. So we predicted that if syrinx size is connected with song repertoire size, we should see similar levels of sexual dimorphism in their syrinx compared to these two species compared to zebra finches. However, we did not find this. We have adjusted male-female ratio on the y-axis and each bronchial half-ring on the x-axis. And in the white is the Korimako or New Zealand bellbird. And compared to the other two species, they had considerably more sexual dimorphism. So this suggests that there isn't a link between sexual dimorphism in syrinx size and sexual dimorphism in song repertoire size. So I then wanted to test whether frequency bandwidth related to syrinx size, but I didn't have the exact syrinx size for each species across these studies, so I used body mass as a proxy for syrinx size. But we did not find a significant relationship. Although it was positive, it was not strong. This suggests that other factors than body mass or syrinx size could be affecting frequency bandwidth, and that might be the individual parts of the syrinx, particularly the half rings. And as you can see, there are quite some outliers within this graph, particularly the female zebra finches and the male bellbird. So the male bellbird has a frequency range that is almost similar to the much bigger European starling. But other studies have shown that there's a relationship between body mass and syrinx size and the frequencies that they sing. And in general, the bigger the animal on the x-axis, they tend to be the lower the frequency that they sing. But not much research has focused on the maximum frequencies that animals vocalize. And in songbirds, it has been found that maximum frequency can link with aspects of body size, such as tarsus length in the male dusky warblers. And why this may relate to syrinx size, if it's a bigger bird, it will naturally have a larger syrinx, so that these bigger bronchial half rings are able to put more pressure on the lateral medial labia to produce higher fundamental frequencies. And so why the male bellbird could be an outlier on that previous graph is potentially their bronchial half rings could be larger than what is predicted for their body size, and perhaps that is allowing them to have a larger frequency range. And so if we go back to these hypotheses, if we see that larger syrinx size predicts higher fundamental frequencies, well, I was not able to show some direct support for that, but the literature is supporting it, and more research is needed to see if sexual dimorphism is a good predictive value for that. And number two, for syrinx size, syrinx sexual dimorphism predicting song repertoire sexual dimorphism, it was not supported by my current research. But obviously only a few species have been studied, and we need to study more species, and I plan to do this for my postdoctoral research, where I'm going to incorporate more species, particularly with female song and within families, the honeyeater family in particular. And I'm going to compare these with more song complexity measures and include other factors such as life history and ecology to really understand what factors have affected the evolution of vocal complexity. And I'll be doing this with the help of new collaborators such as Paul McDonald, Naomi Langmore, Nicholas Friedman, with the help of Australia Pacific Science Foundation research grant. And I'd like to thank all my collaborators and volunteers over the years, particularly the Massey University Research Fund and Marsden Fund grant for funding this research, all the help with the microsetting and 3D modeling, and all my volunteers who helped me in the field. So thank you very much for listening, and thank you very much to the organisers of this conference for inviting me to talk, and I'm open to any questions now. Thanks.
Thanks, Michelle, for this really nice talk and really interesting talk. So while we are waiting for some question on the chat, like I will start by one, I think really naive actually, but and so stop me if I'm wrong, but you analyze only adult zebra finches here, right?
Yes, just adults.
And so my question was like, do you think like if you were analyzing the same in a baby bird, you would actually observe the same sexual dimorphism?
Yeah, that's an interesting question. From what I've read in the literature, it suggests that the syrinx structure is quite well formed already, even as an embryo. So we're not sure yet, but I do have some samples of juvenile Korimako, so I will definitely look at that in the future and hopefully have some results soon.
Okay, interesting. Thank you for the answer. So I have a question from Natasha, who is asking why there will be a bias in the symmetry, right side versus left side.
Good spotting there. Research so far has shown that each side of the syrinx is particularly well formed for producing actually different frequencies. So I think it's the right side of the syrinx that is known more for producing the high fundamental frequency, and the right side is better at producing the lower frequencies. So that's another aspect I'd like to further look into later on too.
Okay, thank you for your answer for this question. So Valentin is asking, is the syrinx size also correlated with volume of the call?
Volume of the what, sorry?
Of the call.
Volume of the call?
Yeah.
I'm not sure what you mean by volume of the call, but
I guess that like the distance that you can cover with your call, like the intensity of your call, I guess.
The intensity, yes. That is not something that I noticed to be measured really so far, but it's definitely something we could look at further. Yeah.
Okay, interesting. So, I mean, we can wait a little bit for the chat to ask question, because as you know, there is a little delay between your presentation and the live. So, like, there's still some question that can happen late. But yeah, to just go up on my last question about if you would have the same dimorphism when they are baby. Actually, just that by curiosity, I was wondering like if you was living a male with only female, maybe like it will absolutely you will absolutely observe the same pharynx in both in the end, in male and female, just because like the male did not have any example of another male to just produce the same sound and finally like kind of train the pharynx. You see what I mean?
Yes, I see what you mean. Yeah, it's an interesting point. Yeah, we're still not sure yet why particularly Korimako have different song between the sexes, but yeah, if they do learn, if they say they did happen to learn a female song rather than a male song, then yeah, we're not sure. I'm not sure what would happen. I guess it depends on how well they copy, but I guess they would be able to still produce higher or lower fundamental frequencies in the females.
I see. Okay, thank you for your answer. So we have time for one more question. So we have Akwashiki Ombugadu, who first say that it was a really nice presentation, and who ask, will your postdoc focus on molecular aspect of vocal complexity in the brain of the songbirds